Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 30th January 2021, and this is something related to practicals. I have been telling you that in practicals you have got four type of questions to be answered. Question number one is the mega question, big experiment, wherein you need to perform an experiment, and that is for 5 marks. And then second question is for 4 marks, one small minor experiment. And third question is you need to take one section, transfer section, TS, that's for 5 marks, wherein your skill will be involved. You need to present a beautiful slide and taking all the precautions to have a clear image of transfer section. And now comes the fourth question, which is very important, and it is related to theory also. That is for 7 marks. Here in this 7 mark question number 4, we give you some specimens and we put some slides under microscope. What you need to do is first identification. Point number 1 is you should identify what is the name of the specimen that is displayed, that is kept in front of you. Second, classify that animal or plant. Classification you have to write. Third, identifiable features. What is that you can identify under the microscope or through naked eye. Fourth, conclusion. Based on that, what is that it is given to you. So these are the things that we expect. And you have this 2 plus 2 plus 3. So out of these 2 marks, it may be one is specimen, another is a slide, and one more maybe specimen. So one plant specimen, one animal specimen, and one slide. In the slide you got for your batch 2 things. One is the Ts of testis, another is Ts of ovary. So if one batch gets a Ts of testis, another batch gets Ts of ovary, and that is for 3 marks. You need not explain, you need to only draw the diagram, identify, label it. But since the slide is related to theory, chapter number 3rd, even 2, so you need to, sometimes if the Viva questions will be there at the end of the practicals, the external examiner may ask you some questions related to the slide. So better prepare theory part of also. So we go with the experiment number 4, that is actually type question 4. In that one, aim to study the discrete stages of gametogenesis, genesis taking birth, gametes, the reproductive cells, in mammalian testis and ovary. So for this, what is required? You need permanent slides of Ts of testis and ovary. Thankfully, we are having both in our lab. And compound microscope, lens cleaning paper, and cleaning fluid, all are available with us. So what is the principle involved? In all male and female organisms, gamete formation takes place in their gonads. Now you must be careful about these two words, gamete and gonad. Gonad is a reproductive organ, like testis is a gonad. Ovary is a gonad because they are reproductive organs or sex organs. What about gamete? Gamete is a reproductive cell or sex cell. Like male gamete is sperm, female gamete is ovum. So don't get confused between gamete and gonad. So gametes, particularly sperms, are formed from the testis, and ova are formed from the ovary. The process of gamete formation is called gametogenesis. Involves meiosis. In the previous session only we discussed. Because of meiosis, haploid from one mother cell which is diploid, 4 daughter cells with a haploid will be formed. So the gametogenic development in testis is called spermatogenesis, and in ovary it is oogenesis. Recollect from your theory classes, okay? And that schematic sketch also, if possible, practice once. They exhibit marked differences and can be examined in the Ts of these organs. So you need to take transfer section. Of course, we have got ready-made permanent slide in our lab. You can do that. What is the procedure? Clean the slide and microscope's eye and objective lenses with the help of lens cleaning paper. using any cleaning fluid. Place the slide on the stage of the microscope and observe first under low power and then higher magnification. Observe various stages of gamete development. Record your observations in the notebook and draw label diagrams. But I have told you procedure, it is in the present tense, but you need to write in the past tense. Next, what is the observation? Ts of testis. Now first to draw the diagram, beautiful diagram always. Seminiferous tubules. Identify spermatozoa, germinal epithelium and spermatogonia. Now you must be having clarity. Spermatogonia, spermatozoa. Now you will observe a large number of seminiferous tubules under lower magnification. Observe a complete tubule in higher magnification and view various stages of gamete development from periphery towards lumen. And identify the following types of cells: germinal epithelium, spermatogonial cells, primary spermatocytes, secondary spermatocytes, spermatids, and finally sperms, that is spermatozoa. So you need to write again in the past tense that you have observed this. Of course, you can observe it also. Okay, under microscope, high power, you can observe. So whatever you observe, you write. Okay, then going ahead with the next one, that is in Ts of testis, the space between tubules are filled with blood vessels and a specific cell type is called Leydig cell or interstitial cells. Now Ts of ovary, draw the diagram first, and when you are drawing it, you must identify various forms of follicles starting with the primary follicle, secondary follicle, tertiary follicle, Graafian follicle. Okay, so in the Graafian follicle, that cavity in the center, antrum, and then the Graafian follicle breaks off, ovum will be released, that's oogenesis, and the broken part is called corpus luteum and corpus albicans, and release of ovum. All these you must identify. So Ts of ovary, there is a mass of tissue lined with germinal epithelium. Inside that you will observe a ovum, which is a cell surrounded by 1 to several layers of follicular cells, as you are seeing. As the ovum matures, the number of surrounding follicular cell layer increases. In the later stage of follicular development, a cavity called antrum appears. So first you must draw double membranous Ts of ovary, transverse section. In that you must draw some blood vessels, and then most important is primary follicle, secondary follicle, tertiary follicle, Graafian follicle, then breaking of Graafian follicle, showing the release of ovum, then corpus luteum. These compulsorily you must show in the diagram. Then the cavity gets further enlarged and the follicle grows bigger. This is Graafian follicle, ready to release the ovum, and the process is ovulation. In the next stage, you may notice a corpus luteum or corpus albicans, which differ from each other and also from Graafian follicle in their features. Discussion. Spermatogenesis is a continuous process after the attainment of puberty, and this is why gamete development and spermatozoa are observed in a single seminiferous tubule. In case of ovary, the follicular development stages are observed. Now you must write point-wise. What is that you have observed? First you have to write the given slide kept under microscope is Ts of ovary. Second you write, it is Ts of ovary because the following features have been observed by me. First, I have observed the various follicles, right? Primary follicle, secondary follicle, tertiary follicle, Graafian follicle. Then you write, lot of blood vessels are observed, and then corpus luteum is observed, release of ovum, the breaking of Graafian follicle, antrum, all these are, you have to write point wise, and hence it is Ts of ovary. And then sometimes the external examiner will ask you some viva questions. So what would happen if meiosis fails to occur in gametocyte? What will happen if meiosis fails? There won't be formation of gametes. At which stage of follicular development is ovum released? Of course, just how you are drawn, that's a Graafian follicle stage. Spermatogenesis is a continuous process. Justify the statement. Yes, spermatogenesis is continuous process in human beings, and once it starts from puberty until the late stages, it goes on. Draw a label diagram of Ts testis. Of course, you have to draw and label it. Draw label diagram Ts of ovary, because that will fetch you 3 marks. Already I told you, compulsorily you practice this so that 3 marks are safe in your pocket. What would happen if sperms are devoid of their tail? Of course, they cannot show movement. Without movement, they are not fit for fertility. What are the consequences of failure of ovulation? Of course, if ovulation is not there, then there won't be release of ovum, there won't be fertility or fertilization or pregnancy. So like this, sometimes the external exam will ask you questions related to this, and be prepared for this. And in this experiment, most important is your skill of drawing beautiful diagram. Okay.